All right, so the next topic is the infected cell. And this topic is probably, you're just going to feel a little bit intensity compared to the other chapters in here. I mean, like this chapter is really intense because there are so many things going on in here. So again, you will need those some detailed information for your quizzes, but I will make sure that I will prepare you well for the exam as well. So what we, if you think about that, what we have studied, we talked about the DNA replication transcription, but one of the key steps that we completely left, that was translation. Now, the translation of the viral proteins is exactly similar to the eukaryotic translation. That's why we did not talk about that. But here we are going to see that how the things are altered. That means that how it suppresses the host of the eukaryotic translation, enhance the viral translation. And along with that, we are going to again revisit the enhancement of gene, enhancement of signal transduction or specific signal transduction, enhancement of gene expression, and then finally the metabolism. So these are some of the specifics that we are going to talk about. So here, we are talking about, the picture is just a reference picture, it's nothing specific to the cell, that we have talked about gene expression, genome reproduction, assembly of the particle, and these depend on the host metabolic, biosynthetic, and signaling and trafficking system. And this is an integrated description of the impact of the virus in an infected cell. Remember that it is not a normal cell, it's an infected cell, and we are going to talk about all these four aspects in here. What are these four aspects? It is the alteration of the signal transduction, inhibition of cellular gene expression, metabolism, and also remodeling of cellular organelle. We just saw that autophagic vesicle, how it remodels those. Those are some of the structures. What you are going to see that it is, so that you can just keep track of it, on the bottom left, you will see that slide what it is related to. Like if it's signal transduction, it will be written as signal transduction here. So we'll start with the signal transduction. So right here, you can see that this is a signal transduction section out of those four. So these would be the four headings or the part of the chapters. So as you can see that the cells must sense their environment and respond appropriately, and this is done by the signaling cascade. It is an extremely complex process, and we are not going to talk about all of the details of that. And even after this complex picture, that this is just the tip of the iceberg that we are going to discuss. So this pathway is called the PI3K, right here, PI3K, AKT, and mTOR signaling. So this is right here, PI3K, AKT, mTOR signaling. So this is basically one of the common pathway the virus actually modulates. And how things happen right here is an idea that the virus can bind to the integrin. Say, for an example, many viruses use, say, adenovirus co-receptor is integrin. It also CAR, coxsackie virus adenovirus receptor. Along with that, it also needs to bind to the integrin. So once this binds to the integrin, it will signal PI3K. Also, some of the viruses will basically cause the receptor tyrosine kinase to dimerize. That can signal PI3K. Also, some of the viruses can activate the G protein coupled receptor, GPCRs. That can also activate PI3K. So the PI3K, or the phosphoinositol 3 kinase, basically phosphorylates a membrane lipid. Remember that, it's a membrane lipid where it makes a PIP3 or phosphoinositol phosphate 345 phosphate. So phosphoinositol phosphate 3 or triphosphate. This basically, on the other hand, phosphorylates another protein which is known as the PDK1. This is a typo in there. This is PDK1. So which is the PI3 dependent kinase. Once this phosphorylation is done, it will activate the AKT, and from here it will do a lot of different things. So let's go one thing at a time. So you can see that the AKT as well as P3K, it also can activate this row family of GTPases, which basically can activate actin remodeling. Why you need the actin remodeling? Remember that when you have a cell, Underneath the plasma membrane, you have a full layer of actin as a cytoskeleton. This is called the cortical actin. It keeps the cells in its shape. If you need to endocytose or bring the vesicle of the virus enclosed in the for the process of endocytosis, if you need to invaginate the plasma membrane, you need to remodel or depolymerize the actin so that this area of the plasma membrane can lose and invaginate inside. So this depolymerization is done by this Rho family of GTPases. And who does this? You can see that these signaling is enhanced by adenovirus, influenza virus, Epstein-Barr, HHV, and these are the different proteins that are involved in that. You do not have to memorize that. Just the overview of that, what these viruses do, is basically the idea. On the other hand, one of the important things that a virus wants to do in the cell, they want to increase the cell survival and increase the amount of cell replication. That means translation of proteins so that it can utilize those proteins. So here, what we are looking at, that the AKT-mTOR signaling, and again, the individual names are not required, see that when they are activating these transcription factors, they are all transcription factors. It enhances mRNA translation, cell growth, and these are also important for the virus. On the other hand, they also are involved in the autophagy. Basically, what happens that they scrap the cellular organelles that are not required. So it also enhances the autophagy and the cell death. But on the other hand, see that what they do, that right here, these two pathways, like one is through the MDM2 and the P53, these are basically the pathways if the DNA of the cell is damaged, because what happens that it enhances the cell cycle, right here, cell growth, cell cycle, and sometimes the DNA is damaged in these cells, and that can lead to these BCL2 backs lead to apoptosis of the cell. And these pathways are blocked by these different viruses. So you can see that these are the herpes simplex, human cytomegalo. I mean, like, I'm not reading each of one of them, and these pathways are basically blocked. So depending upon that what virus is it, they basically alter all of these different pathways. That's the overview of that. So here, one of the important part that we are looking at, it promotes the actin remodeling or depolymerization. It enhances proliferation, cell growth, autophagy, and it basically blocks the apoptosis of so it doesn't allow the cell to die because it's thinking that it is infected with a virus. That's the take-home message from this slide. So here we are looking at the signaling by a PI3K in some individual. These are three examples of that, and this is basically basically in connection with the previous slide. So here we are looking at an adenovirus with the integrin receptor. Once it receives the signal, the integrin, the signaling cascade basically activate this Cas, which basically phosphorylate the SERC, C-SERC, cellular SERC, which activates the PI3 kinase, and it helps a small GTPase RAC, and that basically activates the CDC42, which does the actin remodeling. Again, you do not have to remember all the names, but basically what it is doing, that adenovirus, upon activation of this pathway, it does the actin remodeling, which helps the virus to be endocytosed. Without that, it cannot. So here you can see that adenovirus has two receptors. One is a CAR right here, and this is the integrin co-receptor. And the reason that it needs the co-receptor is to remodel the actin or depolymerize the actin so that the endocytosis can happen and the virus can come inside. The next one is the influenza virus. So it binds to the sialic acid, as you know. So as it binds in there, the influenza virus basically helps in the clustering of the receptor tyrosine kinase. So the receptor tyrosine kinases are in a lipid rafts or in microdomains on the surface of the cell, and they live as monomers, or they stay as monomers. But by the signaling of the influenza virus binding to the sialic acid, these lipid rafts in the area where you have the monomers, they dimerize. And the dimerized receptor tyrosine kinase autophosphorylate themselves, and they now become active. And it activates two pathways. One is a PI3K, and with another protein, FAK, they basically remodel the same way, right here, just another pathway, remodel the actin for the endocytosis. And this pathway is also enhanced by the AKT. And right here, there is also another receptor tyrosine kinase also activate the MAP kinase 1 and 3. The MAP kinase 1 and 3, on the other hand, what they do, that they are responsible for recruiting these proton pumps or the vacuolar ATPases. Definitely underline that. The structure that you are seeing, those are the endosome associated, written as vacuolar ATPases or H plus or proton pumps. What the function of those are, and this is common, this is just not happens in the virus. This happens, acidification of the endosome is a normal pathway. So these will pump the proton inside the vacuole, inside the endosome, and that will basically disintegrate the the membrane and allow the virus to fuse with the endomembrane and release of the genome in there. So this vacuolar ATPase through this pathway is very important. So remember that PI3K basically help in the vacuolar ATPase, which is also the proton pump is basically the same thing, which makes this endosome acidified. Acidification basically is the trigger for the fusion of the virus. There is another one for the Ebola virus. It gets into the cell with a protein, receptor protein called TIM. Once it goes into the endosome, it basically also activates the PI3K via AKT. Now, in the endosome, there could be two different pathways that it can take. The endosome is acidified and it fuses with the lysosome, and the lysosome basically digests a portion of these glycoproteins, which we call as a glycan caps, that basically caps the spike proteins that you are seeing basically on the virus. And the removal of that cap help the virus to fuse with another protein called the niminin-PIK protein, NPC1. We have seen that in the viral entry, and allow the virus to penetrate the endosomal membrane and release the nucleic acid. Now, there is another pathway that can happen where the lysosome doesn't fuse with the endosome, and this forms a cytoplasmic vesicle. This pathway is a non-permissive vesicle because unless the lysosome is fused with the endosome, it will not be able to remove that glycan cap for the virus to enter. So in this process, the activation of the RAC basically blocks the non-permissive pathway where the endosome is not fusing with the lysosome. So this is basically the process. So basically always you have a productive infection and it blocks it. So these are all done by the PI3 kinases, as you can see. So the next part that what we are looking at, this is again, just repeating the process basically for just a little bit more simplified way. So PI3K does have two subunits. The P85, these are molecular weight wise, the P85 is the regulatory subunit and the P110 is the catalytic subunit. So these are both part of the PI3K. And see that a couple of examples. Hepatitis virus NS5A, non-structural protein, binds to the P85. Rotavirus binds to PI3K. Adenovirus E4. And all of them signal through the AKT and through the mTOR, which enhances the translation, increases the cell survival, and basically what it does that it blocks the apoptosis. So you can easily see that these are all favorable effects of the cell. And the cell cannot just kill itself because it's infected with a virus, because it just blocks that process. So these are some of the processes that they do carry out. On the other hand, here you can see that the human herpes virus, they can activate the GPCR, and hepatitis B virus X protein, hepatinoviridity, that also activates or binds to the PI3K with the AKT signaling and blocks apoptosis through mTOR. It induces autophagy, clearing out of the extra or the cell organelle that is not required for the viral replication, increases translation, and see right here we call it cell survival, but basically cyclin D and increase of the cyclin D is associated with the progression of the cell cycle, so cell proliferation. So this is basically, you can think about that right here, these were the outcome in little bit detail. This is a little bit simplified way that what viruses basically does what and where they actually act. So that's basically common activation of the PI3K mTOR relay in the signal transduction. That's pretty much it. We are not going to talk about it anymore because that's good enough for that. So the next one is the inhibition of cellular gene expression in the next topic. So here we are looking at that viral RNA, and this viral RNA blocks AKT activation to induce apoptosis. So how this process is basically happening in here. So this is right here is the genome of the hepatitis virus, and basically what it does, so you can see that there is a 5' cap, and this is the whole polyprotein, but you can see 5' cap, 3' untranslated region. So a cellular protein basically removes so the virus is basically making this genomic RNA. Some of the genomic RNA is utilized for something else. They are not packaged into the virus, but instead a cellular protein with the help of a viral protein removes that cap, and once that cap is removed, there is a cellular protein called XRN1. It chews up this protein, this RNA from all the way from the 5' and all the way towards the 3' UTR. So you can see that it starts chewing. This ORF is the whole thing right here. This is the ORF. So it chews down all the way up to the 3' UTR, and right here there is a certain sequence that basically blocks the XRN, these secondary structure, and this basically produces this what is called as sfRNA, or sometimes called the sgRNA. sfRNA is basically subgenomic flaviviral RNA, or if you tell an sgRNA, you can talk about the sgRNA is a subgenomic RNA. Basically, it is not an mRNA because we are not talking about subgenomic mRNA, but it is just a chewed up small segment of the RNA. That's why we call it as a sgRNA. And what happens that this sfRNA inactivates the AKT and promotes the apoptosis basically at the late stage, basically killing of the cell. And this is, so remember that. Sometimes the virus wants to kill the cell, sometimes virus wants to keep the cell alive. So it is a balance. In this case, we are looking at that after the viral replication at the late stage, they are, because remember that the first genome replication doesn't happen. It transcribes, it makes the proteins, and all things are done at the very end of the cycle. When the genome replication is happening, viruses are packaged. At that time, it wants to lyse and kill the cell. So this phenomena is happening at the late stage, and what you are seeing here, that in the wild type, these are the plaques, basically, where you see the viral replication. But what you do, that if you just mutate it, this particular virus, in the mutate form, you cannot see any of these plaques. So basically, it just doesn't allow the virus to kill the cell. So in that case, you just you cannot produce any sfRNA basically. So if you cannot produce any sfRNA by deleting the specific XRNs or remove that, the concentration of the anti-apoptotic protein remains constant, and it cannot just kill the cell. So basically, if it can produce the sfRNA or the small flaviviral RNA or subgenomic RNA, it induces apoptosis. Without that, they cannot induce apoptosis. So this is a very unique process. So after that, we are looking at many different cases, and again, there is no need for me to write those. And again, the thing is that you will be asking that what is happening in all of these cases. We are going to just talk about some of the specific ones. But you have seen this, that right here at the very level of transcription, polyadenylation, splicing, export, translation, degradation, all of these things are happening by different viruses. And we are going to take some of these examples and talk about it in the next part. So And these are all basically to inhibit the gene expression of the host cell. So here we are looking at first some of the export of the cellular mRNAs out of the nucleus into the cytoplasm. So basically this is the suppression of the host gene expression. You need the mRNA of the host cell to be transported for them to be translated. So how they are doing this? Say for an example, for HSB, ICP27, they block the splicing. So blocking of the splicing of the mRNAs, that will basically cause them to stay inside the nucleus. They cannot be exported. Then this adenovirus E1A, E4B, these proteins associated with all these different subunits, which are basically E3 ubiquitin ligase, they basically, so what they do, that they basically ubiquitinate protein responsible for transportation and splicing, and they will basically degrade. Remember that the function of the ubiquitin ligase is basically to add ubiquitin to the protein so that they are basically degraded by the proteasome. So these proteins basically, the proper mechanism is properly not known. What is their function in the export? But these proteins, which are expressed in the nucleus, or transported in the nucleus back, these are viral proteins. Remember that they are produced and then transported back in the nucleus. They are associated with E3 ubiquitin ligase. They are transported outside, and they basically degrade many of the host proteins in there. So on the other hand, here you can see that this is a poliovirus and human rhinovirus. They contain these proteases, 2A-pro. This is a 2A proteases. They basically chew up this NUP62, which is an adapter protein for the export of the mRNAs. So the mRNA, if they are not degraded, they are great. Right here, you can see that this is the spliced mRNA that is with the adapter protein. They should be able to be exported into the cytoplasm. But if these NUP62 adapt or 153, these are degraded, this will not, the nuclear pore will not be able to recognize this, and the transportation halts. It will stay inside the nucleus. On the other hand, here you can see that same way that some of the influenza virus NS1, they sometimes hyperphosphorylate some of these protein that cannot transport. Sometimes they bind to the poly-A binding proteins, and that's also modifies the polyadenylation, and that also blocks the transport of or export of these things. And VSV also blocks the export. So these are some of the ways that inhibition of splicing and just the different mechanism that how the export of the cellular mRNA is basically halted. The next one is which we call as a mRNA turnover. So in this process, you are looking at that this is a normal process. Remember that this is a normal process of the mRNA decay in a cell. This happens in the regular cell with the regular mRNA. You have the mRNA, once they have produced enough amount of protein, when they are not needed, how they are degraded. They basically bind to one of the proteins, which basically is CCR4 associated and NOT or PAN, and these complexes do the polyadenylation or deadenylation. They remove the poly-A tail, CCR4 NOT PAN. And after this deadenylation, they can go into two different pathways. So one is this DCP1, which is basically activates DCP2 with the help of HEL-S. These will decap this 5' cap. This can only happen once this deadenylation is done. So these are the decapping enzymes. And so the cap is removed. Once the cap is removed, XRN1 that we have seen, that they can chew up the RNA from the 5' end. On the other hand, what can happen, the 3' to 5' end, which is from this side, this can happen also by the exosomes. These exosomes are the structures with endonucleases and exonucleases, and they can also degrade. And also there is another protein, DCPS, that can also cleave from the 5' end. So even without the decapping, the RNAs can be degraded from the 5' end, or they can be degraded from there. So what happens, poliovirus RNA when they are making the mRNA, that these proteins will also try to decay them. So what they do, these are degraded, the pan-DCP1A exorin, these are degraded in the poliovirus infected cell. So the viral mRNA cannot be degraded. Now the question will be, okay, all right, so if they are not degraded, how can, so then the cellular mRNA is not degraded as well. So what is the advantage? Okay, all right, so here, right here you can see, poliovirus will to take out this with a 2A protease, the NUP153. So basically what's happening, the cellular mRNAs that are to be transported, they are not being transported for this, they are stuck into the nucleus. Poliovirus mRNA with the RDRP is produced in the cytoplasm, no need for export. But there is no cellular mRNA, but you have poliovirus mRNA in the cell, and that is basically preserved because the mRNA decay enzymes in the cytoplasm are degraded. So this is basically the logic that how the virus channelizes itself to make or protect its mRNA, increase the gene expression, whereas shutting down the host. So here there are several proteins that initiate the mRNA degradation, and of course this is what we call as a host mRNA degradation. So for example, right here is the cap binding complex of the translational machinery, and this is M7-GPP-NP, this is the cap, and this is the poly A binding protein with the poly A tail. So these are just example of HSB VHS or say HHP SOX. They basically chew up this cap binding complex and they destroy these caps, and as they destroy these caps, you know that XRN1 basically right here, XRN1 will chew them up, and the SARS-CoV-2 basically does the same thing as well. So here you can easily see that the other proteins like SOX or NSP1, they can also cut or degrade the DNA. The VSCV, varicella, like vaccinia virus as well, not varicella, vaccinia virus, also D9, D10 protein, they can also decap, and you can see that this is the same process, that as they cut these specific areas, cut out the caps, these specific proteins, XRN1 will degrade the mRNAs that are present, and these are the host mRNAs. Now we are going to see that how the viral mRNAs are going to withstand this. So we're going to see this. So here, on the other hand, is a HCV genome and a microRNA, which is the myRNA 122. This is required for the HCV replication. And see that how this myRNA basically blocks the 5' UTR or the untranslated region and blocks the degradation by XRN1. So basically here we saw that the cellular mRNAs are being degraded. And here a process that we are seeing where this viral RNA doesn't have a cap. Remember that HCV doesn't have a cap. And how this is basically being protected, this miR-122 mRNA or microRNA binds to this 5' end, and that will not be chewed up by the by the XRN1. So if you target miR-122 you can make a silencing product for this one, and if you can do the silencing of this miR-122, that has a therapeutic value for the HCV that are being investigated. All right, so here we are just looking at the modulation of the translation, because all viruses need to make mRNAs, and again the mRNA of the virus will be used for the translation. So what are the different processes? Now, this slide has nothing to do with your exam or the quiz. This is just eukaryotic translation, just a quick recap. You do not need to memorize each and every factor, but just the core value is important. So here we are looking at how the initiation complex brings in the 2 subunit of the ribosome in a regular eukaryotic translation. So you have a 40S subunit and say EIF1A, which is eukaryotic initiation factor. EIF all stands for eukaryotic initiation factor. 1A and 3 binds to the 40S, and then a ternary complex. So just remember this ternary complex. This word is important. So this ternary complex is basically you have a tRNA and with a methionine. So this is a methionine tRNA with a methionine. Then you have the bound EIF2 GTP. This is your second. So first one in the ternary complex means there are 3 things in there. So one is your tRNA with the methionine, second is EIF2 GTP, and EIF1. These all together is basically the protein, and say that UAC this is the anticodon which will be bound to the AUG. So this is the anticodon of the tRNA. So that's the same part. This is nothing different. So tRNA, EIF2 GTP, EIF1, 3 things. These are recruited onto the 40S subunit. You can easily see right here. So which is already the EIF1A, EIF3 is together. And now comes this EIF4G and another protein EIF4A, which we call as a scaffolding protein. This EIF4G is very important. So just underline that thing. The ternary complex, as I said, EIF2 GTP, that was important, and this EIF4G. So now what happens that this complex will recruit this EIF4E, which recognizes the cap. So EIF4E recognizes the cap. So recognize this EIF4G, and also this is another one, EIF4E. This is also cap binding. This underline this one as well, EIF4E, which is a cap binding initiation complex. Now at this time what happens, with the help of ATP, this complex with the 40S subunit of the ribosome will go back and forth and scan the beginning of the mRNA. See that right here, AUG, it will go back and forth, and it will find the AUG, and now you can see the anticodon of the epithelial tRNA UAC is sitting on AUG. Once this structure is formed, see that EIF5 comes in, and this EIF2 bound with the GTP is hydrolyzed to GDP, and all these factors go out. And all these initiation factors go out, and the 60S subunit is recruited, and that will be the start of your translation, basically. So this is basically the assembly of the initiation complex of the ribosome. So what we are going to see that how this process is perturbed in the cell. So here, this is just an example of a real-life experiment. And just remember that we can use some of these pictures for your exams. So determine that what we are looking here. So if we just take some cells, and in fact with the poliovirus that has been used in the experiment, we are looking at the time post-infection, 0 time. We have infected the cell right here, 1, 2, 3, 4, 5. We are taking out cell extract. And this is the rate of protein synthesis. Okay, protein synthesis in the cellular protein synthesis is high. And gradually you can see that there is a shutdown of the host protein synthesis. Host protein is not being produced anymore. And there is a rise of the poliovirus infected cell in the protein synthesis. These are all poliovirus proteins. And then it will come down because the cell is being lysed and the viruses are getting out. So if we just take in this 1, 2, 3, 4, 5, we take the samples, these are all cellular proteins. These are also cellular proteins. Now, after 3-hour post-infection, see that some of these bands of proteins are getting fatter. Here, the background is gone, and you have pretty much all these bands are the viral protein, 3CD, 1CD2, BCVP0, 2CVP3. These are all different viral proteins. So you can easily see that how the cellular proteins, all these are cellular proteins, they are shut off, and just exclusively the viral proteins are made by the 5th and the 7th hour of infection. So this exactly illustrates the process. On the other hand, you can see the protein synthesis in uninfected cell. They are just a straight line, no effect. So this is what we're talking about. That is why we talked about that EIF4G and the EIF4E. These are the 2 important factors in the initiation complex, and how the modification of these actually helps in shutting off the host protein synthesis. So the first one, we are looking at the poliovirus 2A. It cuts out the EIF4G. right here. It will just choose it off. So EIF4E cannot be recruited. No initiation complex. This falls off. On the other hand, see right here, adenovirus, influenza virus right here. This with EIF4E dephosphorylation, this is a phosphorylated EIF4E. That is absolutely needed for the binding. If it dephosphorylates right here, there will be no binding. On the other hand, there is also another protein which is called this 4EBP1. This 4EBP1 is phosphorylated. When it is not phosphorylated, it basically binds to EIF4E. So this is basically an inhibitory protein in the cell that blocks, when necessary, the cellular replication. But you can see that this particular poliovirus and encephalomyelitis virus, the viral protein dephosphorylated 4EBP1 that binds to EIF4E, it no longer can bind to EIF4G. Because this is basically the cap dependent translation. You need this complex to be bound to EIF4G4E to recognize the cap. And everywhere EIF4E is not being allowed to bind to this. So how the virus survives in this case if it doesn't bind the cap? So here is what it is, what we call as the iris, internal ribosomal entry site. These viruses don't have a cap. They are recognized by the iris. And this is the beauty of that thing. See, look at that. Right here, hepatitis A virus iris requires all of these regular machinery. So it does not take up or it does not degrade EIF4E or EIF4G. On the other hand, in a type 1 or 2 iris, we're just going to see that, just say the poliovirus, they chop off this EIF4G. It does not bind the EIF4E. So we just saw that right here. So this is chopped up by the poliovirus. And the poliovirus iris can be recognized just without the cap recognition site. These secondary structures are recognized. Hepatitis C virus, it does not even need any of these structures. Just the 40S subunit and EIF3 can recognize its iris. So these are basically the different ways that even the degradation of the translational initiation machinery can be bypassed in the viral replication itself. So here it just shows the internal initiation of the poliovirus, reminds you that this right here is the internal ribosomal entry site. And here it just gives you, again, nothing, no questions from here, just gives you the type 1, 2, 3, 4, 5 iris. And here is just an idea that where EIF4G basically binds. So you can see that there are many different ones. The AUG sometimes are downstream outside the iris, except right here, you can see that the AUG or the start codon is inside the iris. And here in this type 4 iris, there is a non-AUG start of the translation, which is very rare, non-AUG start. The first amino acid is always methionine and coded by AUG. And here you can see that right here is an alanine start right here with this. And here the AUG is inside the type 4 the type 4. Both of them are type 4. That AUG sits inside the iris basically. So let's see that. We'll stop here and we will just start from the initiation of translation inhibition from here in the next class. So we are just about halfway through already. All right, so we are still at the infected cell, and we'll finish this and then we'll try to do the next chapter. We'll start the next chapter as well. So we'll finish two of the chapters this week. So we will have 2 quizzes due this Sunday, and then we'll finish the other one half. So that's pretty much it. So what we are looking here, this was the overview of the eukaryotic 5' dependent translation initiation complex. And here I just wanted you to focus on this ternary complex, which is essential to bring other factors on top of the 40S subunit, which is a ternary complex. Why you call it ternary? You have this EIF, eukaryotic initiation factor 2, and along with that you have the GTP, charged with the GTP, EIF1, and this methionine first. This one is the tRNA bound to methionine. This is the ternary complex. And this is important, and then we are going to talk about this, that how this is affected. So we all covered that, and here what we are looking at, that upon viral infection, the first thing the cell does after sensing that the viral infection has occurred is a global shut-off of translation. It just shuts off the cell so that the cell will die, and it will also kill the virus along with that. So that's a global shut-off. So this slide basically shows how the global shutoff basically happens. So here you are looking at this EIF2 right here. And this EIF2 has basically 3 subunits. It's alpha, beta, and gamma right here. And they are not shown here, but it's alpha, beta, gamma. And the EIF alpha does have a phosphorylation site. So again, remember that, alpha, beta, gamma, 3 subunits, EIF2. And the alpha can be phosphorylated. But here in this complex, which is coming out of the ternary complex with the GDP and EIF2, this is not phosphorylated. Now, this structure, this initiation factor with the GDP, is captured by this EIF2B. This EIF2B exchanges the GDP with a GTP. As soon as the GTP replaces the EIF2, it is charged again. EIF2B goes out, and this helps the ternary complex formation, which is the tRNA, EIF2 bound with the GTP. And again, you know that that will be recruited on the 40S, EIF1, EIF3, all other things will come off. So as you can see, this ternary complex is basically essential to form the ribosome. Otherwise, the translation will not go on. So what it does, that the cell, without viral infection, there are some activators. Say, for example, ER stress. These are cellular stress without viral activation. In the ER stress, there is one kinase. This is a PKR, protein kinase R, like ER kinase. Amino acid deprivation induces a protein called GCN2. And if it senses double-stranded RNA, this is only double-stranded RNA can be sensed only upon viral infection. There is no other double-stranded RNA state. Then it activates PKR, protein kinase R. So these are all phosphorylating enzyme, and what it does, these are all EIF2 alpha kinases. They will take this EIF2 GDP and add the phosphate to its alpha subunit. As soon as this EIF2 is phosphorylated, it binds to EIF2B in an extremely tight manner, very high affinity. So both words are important. It tightly binds with a very high affinity, and it basically, as you make more and more EIF2 phosphorylated EIF2, it scavenges out all these EIF2B that is in the cell. So this ternary complex formation is stopped. This is the global translation shut off, basically. And then eventually, if the cell survives, there are some phosphatases that can take that phosphate off to release the EIF2B, but that is we are not discussing. But this is the mechanism of global translation shut off. So this is the understanding. Just remember that unphosphorylated EIF2 GTP-GDP exchange can happen. On the other hand, if EIF2 is phosphorylated, it binds to EIF2B in such a tight manner that it cannot be exchanged, the GDP cannot be exchanged with the GTP, and it scavenges out all the EIF2B and halts the initiation complex formation, basically. So what the virus actually does, we'll just come to that. So now we talked about the double-stranded RNA induces PKR. That basically does this EIF2 alpha kinase phosphorylation. So this is how we are talking about antiviral mechanism. So this is the antiviral mechanism, and this is the protein kinase R right here in the second line here, inactive protein kinase R. And they have a kinase phosphorylating domain, and there are 2 domains. These are the double-stranded RNA binding motifs, 1 and 2. This PKR can be activated by two mechanisms. One is by the PACT3 domain, PKR activating domain 3. If this protein binds to PKR, this can be activated. On the other hand, if there is a double-stranded RNA present, these double-stranded RNA binding motifs bind to the double-stranded RNA, and these subunits cross-phosphorylate, cross-autophosphorylate to each other. Very carefully remember that that's always an exam question, that this PKR activated by the double-stranded RNA cross-autophosphorylate each other. So basically what we mean by that this kinase domain will phosphorylate the other one, this kinase domain will phosphorylate the other one. Now you have an active PKR. What is the function for that? The function is the global shut-off of the translation. So this is your antiviral mechanism. So why we are talking this? We are talking this because we are going to see in the next slide that how this mechanism is halted by the viral proteins, so or the RNAs. So here is one example. For adenovirus, actually produces these virally associated RNAs. These are produced by RNA Pol 3. If you remember from the very back, RNA Pol 3. They are not mRNAs, virally associated RNAs. And these RNAs basically binds to the PKR and make them inactive. So no phosphorylation of eIF2 alpha, active protein synthesis, because active protein synthesis is required for the viral active protein synthesis is required for the viral replication in the cell. So if it is shut off, it's not going to work that. So this is the RNA-associated adenovirus, virally associated RNA-mediated inactivation of the PKR. This is one example. In this one, we are seeing a number of different proteins. And again, you do not have to remember all of them. Maybe if you see a specific example in your quiz, that may be important. So just remember those for the exam, and I will review that as well. So but not all of them you have to remember or memorize. So you can see vaccinia virus K3L, herpes simplex virus gamma 34B. They shut off basically this whole process. Here, the PERK, this can also phosphorylate the eIF2 alpha, PKR, and these are the number of different viral proteins which all basically doesn't allow these proteins to be phosphorylate the eIF2. And vaccinia virus K3L and virus simplex, this directly works on that. Pseudo-substrate means that when these proteins which are phosphorylating eIF2 alpha, this vaccinia virus K3L binds to the eIF, and this is basically the enzymes try to phosphorylate this protein instead of eIF2. And this herpes simplex virus gamma, on the other hand, has a phosphatase regulatory subunit, which takes out the phosphate directly. Even if it is phosphorylated, the phosphatase activity takes the phosphate out, so the eIF2 can go and exchange the GDP to GTP to form the ternary complexes. So these are the processes by which how the viral proteins and RNA in here, via RNA, counter the inactivation of eIF2 alpha. All right, so after that, we are looking at the last part of this. So there are two types of non-membranous granules that is formed in the cell when they are under the stress. One is called the stress granule. These stress granules are usually, most of the time, formed around translation complexes, So you can see that yellow part, this is the translation complex. You can see that this is basically the cap binding protein, eIF4G, and of course, all other machinery. So these stress granules are formed along with the mRNA with those. On the other hand, what is not shown, there is another type of granule which is called the P bodies or processing bodies. Those basically has two functions. It takes up all the RNAs. It usually doesn't accumulate the translation complex, rather it takes out the mRNAs, and the non-essential mRNAs are degraded in there. And some of the mRNAs which are required for the survival of the cells are protected in the P bodies. But just remember that the stress granules as well as the P bodies, these are basically stress-related. When the virus is infected in the cell, it protects some of these mRNAs for the survival and basically inhibits the translation of the cell. So this same function. Once they are inside the mRNAs or inside the P bodies, they are not going to get translated. When the translational complex are inside the stress granule, translation is not going on. So this is also shutdown of the translation. Now, with this stress granule, there are a couple of proteins which are associated with that. These are which you call as a marker proteins, like G3BP, TIA1, and TIAR, RAP55 as well. So these are the marker proteins. And what is being shown basically here, that the number of different viruses counter the formation of the stress granule. So again, know the logic. Stress granules are formed to shut down translation and protecting the translation complexes in certain bodies. And the virus doesn't want to shut down the protein synthesis, so they somehow interfere with the stress granule forming proteins, basically. So let's see at a time. So RAP55 is also another one, although it's not in here. Influenza virus NS1 basically binds to it and sequester it, and it cannot form the stress granule. On the other hand, here you can looking here, right here, that also hepatitis C virus, there are lipid droplets and many different proteins. It binds to G3BP. This is the stress granule making protein that is sequestered in here. The formation is disrupted. Poliovirus 3CBP, on the other hand, see that it just basically cleaves the G3BP, so it cannot form the stress granule. On the other hand, here you can see that the same way TIAR or G3BP, they bind with all these different, again, you do not have to memorize any of the name, just understand the overall function, that they will bind to these G3BPs and they cannot be recruited to form the stress granule. And here you can see that these are all basically bound to proteins or proteolytic cleavage is happening. Here, the G3BP or the TIAR, they are bound to the secondary structure of the virions. And this is a flavivirus. You can see that just like the hepatitis C virus, it's a flavivirus. And the double-stranded RNAs can also sequester these G3BP or TIAR. So all of this mechanism, either protein or secondary structure of the viral RNAs, basically inhibit the process of stress granule formation and doesn't allow the cell to stop translation for their own benefit, because they need to make proteins for the viral replication. So that's about it. So the next part that we are going into is the metabolism. So in the metabolism, hold on. No, this is just for the timing. So in the metabolism, here what we are looking at, that this is the Krebs cycle that you all know. And what we are going to see that this Krebs cycle, not only that, the glycolysis as well as fatty acid synthesis, everything is modulated by different viruses. So basically what happened, that the only take-home message, because we will come back to that one of the virus, say this is the cytomegalovirus, this is a beta herpes virus, HSV1, like herpes simplex virus 1, which is an alpha herpes virus, they tap out different material from different points of this Krebs cycle. So here it takes out the citrate for fatty acid. Here it takes out aspartates for pyrimidine purine formation. So if they tap out at one point, what will happen? The cell need to metabolize and the cycle should stop. But what they do very carefully, they channelize other material like glutamine and just inject, they break down the glutamine and make it to alpha ketoglutarate so the cycle can continue. So it taps out material from one point and also modulate and tap in material in there. So we are going to see these with specific examples. So here also one of the overview of that, and this is done with the adenovirus, wild type as well as a deleted E4 protein, is that what you are looking at some cell culture Petri plate, And these are not infected. These are all red, and these are added with the phenol red, which is the pH indicator. Neutral pH, it's there red, and if they turn acidic, they are going to be yellow. So this is an indicator in the media of the cells. So these are infected with the adenovirus wild type, and see that within 48 hours they turn yellow. That means there is glycolysis, enhanced glycolysis. It produces the lactic acid. That's why its pH changes. If you take the delta 4, or if you delete the E4 gene, which is responsible for the replication of the adenovirus, you can see that there is no effect on that. So that is just been plotted in here. So you can see increased glycolysis. This is basically needed for the production of, or basically the energetics of the whole cell is changed. So here we are looking at the glucose metabolism, and again, you do not need anything in this pathway, but just seeing here that once glucose enters into the cell, you can see it go to the glycolysis steps, but there are two steps. See right here, the glucose 6-phosphate can be converted to ribose 5-phosphate, and this is needed for the synthesis of nucleotides. On the other hand, the pyruvate is converted to acetyl-CoA, and this can be used as the precursor of fatty acid synthesis. So what happens that we are going to see the viruses which does replication of the genome very fast, they want to tap out from here for the replication because they need the precursor of nucleotide to make their genome. HSV actually does that, human herpes simplex virus. On the other hand, there is another, the HCMV, that we were constantly talking about, the human cytomegalovirus, which is also a herpes virus, beta herpes virus. They actually do have a membrane which is very complicated. This membrane is the envelope which contains specialized lipid, very long chain fatty acid lipids and very specialized membrane. They need a lot of protein synthesis and they grow very slowly. They do not tap out anything from here, but they tap out from acetyl-CoA to make their membranes. So that's basically the whole process is perturbed in the virus-infected cells. So let's take a look at some of the specific ones. This is just the two herpes virus, different effects on glycolysis that we were talking about. Let's see that HCMV, it allows the whole glycolysis pathway to go on to acetyl-CoA. From here, it will tap out for the synthesis of the membranes. Whereas HSV replicates very fast in the cell, it needs to make a lot of nucleotides, so it taps out glucose 6-phosphate, and which can be converted to ribose 5-phosphate from this, and that's the sugar that is needed for the nucleotide synthesis, and it blocks the rest of the glycolysis. So, depending upon the virus, it can modulate or perturb the normal energetic metabolism of the cell. That's basically what it is. So here we are looking at also the example that is done by the HCMV or the cytomegalovirus. So here the pyruvate enters the mitochondria from the cytoplasm and its acetyl-CoA is formed. Now acetyl-CoA is required by the HCMV to produce the fatty acids. But acetyl-CoA is such a large molecule, it just cannot just get out into the cytoplasm. It needs to stay in the matrix of the mitochondria. So the next reaction is the formation of the citrate, and the citrate, the carboxylic acid transport protein can transport citrate across the plasma membrane. And see that what is happening, the ATP citrate lyase basically reforms this acetyl-CoA and oxaloacetate, which can again enter into the mitochondria through pyruvate. But this acetyl-CoA is converted into malonyl-CoA, and the fatty acid synthase, a bunch of different enzymes, are going to make the fatty acids. So basically what it is doing, that the Krebs cycle, if we just go back to this one right here, this whole cycle is supposed to happen in the matrix of the mitochondria. But what is happening in this one, HCMV channelizes this acetyl-CoA to citrate and takes it out of the mitochondria to allow the synthesis of its membrane, which is basically the fatty acid synthesis. So that's what it is. So the rest of the cycle is stopped here. But we are going to see that this will be given back to the Krebs cycle at a certain point. Here's another example that the hepatitis B virus, which is the hepatinavirinae, and the HCV, which is the hepatitis C virus, that's a flaviviridae family. They both are implicated into type 2 diabetes, which means that they are insulin independent type 2 diabetes. What they do, that both of them actually help in the gluconeogenesis. What you mean by that, gluconeogenesis is basically formation of glucose from 2 carbon compounds, basically from pyruvate, say. So think about this way, that the glycolysis goes on the other side, I mean, like in a very crude way. So that's gluconeogenesis is basically basically enhanced in here. So basically what happens, that the molecules are already tapping into other organic molecules to make the glucose. Now you also have a dietary glucose, so you basically have a very high blood glucose level all the time, and this is insulin independent. And they also sometimes interfere with the channeling or modulating the glucose transporters as well. So that's basically what it is the liver is the site of the glycogen. So when the glycogen is broken down into glucose, this happens when you are under stress, when you're working out, that basically happens. Also, under stress, this de novo glucose synthesis happens, not in a normal time. But in the infected patients, this broken down of the glycogen, which is glycogenolysis, that is the formation of glucogenesis, and also the de novo glucose synthesis, which is gluconeogenesis. Both of these processes are enhanced in HBV as well as HCV infection. So that's basically the process. So here we are looking at the same thing that this is just a repeat of that HCMV takes out citrate at one point and it gives back the alpha ketoglutarate at a certain point. Also, the vaccinia virus does that as well. Vaccinia virus also use the glutamine to allow the Krebs cycle to go on. And vaccinia virus also need a lot of membrane synthesis, so it also taps out just like HCMV as well. All right, so here we are looking at, on the other hand, the lipid metabolism. So we have seen modulation of glycolysis, modulation of Krebs cycle, glucose metabolism. Here we are looking at lipid metabolism. So most of the lipid is in triglycerol, like glyceride or glycerol state in our body, and they are carried by the lipoproteins, which are the low-density lipoproteins. They are covered with an apolipoprotein, which makes them soluble, because in the blood you cannot transport the lipid droplets. I mean, like, they need to be packaged with a soluble protein. Otherwise, they will, if they come off, they're going to precipitate. So when these molecules are close to the membrane, the lipases can break down them into the fatty acid, and these free fatty acids, FFA is free fatty acids, can be transported inside the cell. With the acyl-CoA, they can enter the ER as the triacylglycerol. So TAG is the triacylglycerol. And this tag is associated with a protein, and these are basically coalesced together to form the lipid droplets. And again, remember that these lipid droplets in the cytosol, which is aqueous, is not a favorable condition. So what happens that it is coated by this perilipin to make them stable in the mature lipid droplets in the cell. These are used by some viruses for their own, for the budding of the viruses. These lipid droplets are used. We are going to see this process in a sense. And also along with that, so we are going to see that for the membrane synthesis is required for the lipid. That's why lipid metabolism is modulated. And also the beta oxidation, which is the breakdown of the lipid, is also minimized in some of the viruses, because you want more of a fatty acid synthesis or fat synthesis, but less of a breakdown. So all of them are basically perturbed in different cases. So here we are looking at that the HCMV, which induce a very long chain fatty acid synthesis for its assembly. What we mean by very long chain fatty acids is that most of the fatty acid that we see in our plasma membrane, they are in between 14, 16, 18, 20. Usually that's the normal size of the fatty acid carbon length, carbon chain. Whereas we have seen in HCMV, they can be as long as 26 carbon. These are very long. It's all in your note. So not only the HCMV require high amount of membrane lipid, but also a different type of lipid, very long chain. And see here, we are looking at that how the HCMV infection increases the carbon flux from glucose to acetyl-CoA. So how basically this happens? We so far says that this happens, but it has to modulate the enzymes in the cell that is doing that function. It just cannot just go and tell that, okay, you go here, you go here. There has to be a reason for that. So here is the way that it modulates these processes that we have just discussed. So here we are looking at an endoplasmic reticulum. So here you have, these are all, this INSIG1, SCAP, SREBP1P, these are all transmembrane protein. They are produced in the ER and they are staying in the ER. So you can see that SREBP right here. These are transcription factors, but this SREBP1, this is a precursor, which is associated with the SCAP. This is basically will activate the SREBP later. And also, this is an insulin-based INSIG1 is a factor. They are all associated together. Now, when in the membrane, like in the ER membrane, you have high cholesterol, like a normal cholesterol, they remain in this state. They remain in this state. They are inactive form. They are all together in here. But if you have less amount of cholesterol or the cholesterol amount goes down, then that is considered as a stress, and this PERK that we talked about earlier, protein kinase, ER-like kinase, they dimerize. But the same way, not without ER stress, when your cholesterol is normal, HCMV basically modulates the PERK dimerization and activation. So what's happened when PERK is dimerized and activated, and this is by the phosphorylation, it basically binds to INSIG1, and this allows these SREBP1, which is a transmembrane protein, to form vesicles, and this is a COP2 vesicles. These are formed in the secretory pathway, and these vesicle, which will have the transmembrane SREBP on its membrane, now comes to Golgi. So this protein is secreted from there. Secreted means, like, remember that. It is associated with the membrane. It's associated with the membrane. It will just come through the vesicle. The vesicle will fuse with that. In the Golgi, as the pH changes, the SCAP will again convert this SREBP precursor to SREBP1 matured. This is the transcription factor that will cross the nuclear pore, and this will bind to specific gene sequences for fatty acid synthesis and sterol synthesis. So this is basically the process of the transcriptional activation by HCMV, and this is how it taps or channelizes, because earlier those enzymes were less. Now those enzymes are more, so it just channelizes the process towards it. Also, there is another protein which is called the lipin-1. This basically blocks the translocation of SREBP1M into the nucleus. This is also by a mTOR pathway, HCMV. We have already seen the AKT-mTOR pathway. This signaling cascade also inhibits the lipin-1, which is the inhibitor of translocation of SREBP1 into the nucleus. So this is basically, and once these are done, HCMV got the genes for the fatty acid synthesis. So that's your transcriptional activation by HCMV. All right, so here we are looking at that in the HCV, this is hepatitis C virus, not HCMV. So here we were talking about HCMV, which is a human cytomegalovirus, herpes virus. here we are looking at a flavivirus, hepatitis C virus. This also produce or induce lots of fatty acid synthesis. So basically what happens that FOXO1, this is a forkhead box protein, FOXO1 is a transcription factor. FOXA2 is also a transcription factor, and they have specific targets. So FOX1 basically induces some specific genes that are basically induced the SREBP formation that will basically form the fatty acid synthase formation and increase lipogenesis. So what happens that normally, think about this way, that if insulin is basically when you have high blood glucose level at that time, you have insulin secretion, and when you have insulin, the insulin basically takes out the FOXO1 or transport into the cytoplasm. Why it is so? Think about this way, that in a normal cell, if you have enough of glucose, you do not need that much of lipogenesis. You do not need to synthesize fat, which can be stored, because you have ready energy. The fat synthesis is required at a point when you have too much of glucose at that point. So that's basically what the normal function is. But that is not required. But basically what happens, that the HCV blocks the transportation of FOXO1 into the cytoplasm, and FOXO1 stays in the nucleus and basically increase the lipogenesis. So that's basically the overall process. On the other hand, what happens right here, that the FOXA2 is responsible for genes for fatty acid oxidation, beta oxidation, which is the breakdown of the lipid. The HCV also downregulates, so this arrow is downregulate FOXA2. So it basically, there are some of the HCV protein binds to FOXA2, and the transcriptional activation of MCAD or SCAD, these are responsible for expression of fatty acid oxidation enzymes. These are downregulated. So you can see that in an infected cell with HCV, this is a mock, means that these are just the normal cells, and the green parts are the lipids in the cell in hepatocytes. And here you can see that how much increase of lipid synthesis is happening in these cells, in infected cells. So HCV severely promote HCV severely promote the lipogenesis, whereas it just downregulate the fatty acid oxidation. So that's all about metabolism. Then the next part, the last part we are looking at, that in the infected cell, is the remodeling of cellular organelles. So here is an HCV example of that why that much of membrane is required for that. And it is for this purposes. You can see that the ER membranes are remodeled by these different structures. Some of the structures are stalk-like structure inside which the virus can replicate. They sometimes also form these multi-membrane vesicles, double membrane vesicles. We have seen it coronavirus as well. Some of them can be, some of the viruses can be replicated on the surface of the membrane as well. So these are the different viral particles. So what happens also, that once these viral components are all there, and you can see that right here this is the part of the ER, and you have the viral envelope glycoproteins in here. Now, once there is enough amount of this capsid protein of HCV is produced, this blue one is the capsid protein right here, what happens that a lipid droplet that we talked about, that it enhances the fatty acid synthesis around that lipid droplet, these proteins, which are the NS5B and the capsid protein, all the blues are the capsid protein. Of course, the perilipin is there. Perilipin is not shown, but that is replaced by these capsid proteins. And on top of that, you have another protein, which is a non-structural protein, NS5B. And all these proteins basically help the formation or budding of the HCV into the ER. Remember that we have seen that the viruses assemble and exit from the plasma membrane. Let's say for HIV, it assembles on the plasma membrane. influenza virus assembles on the plasma membrane. Here, HCV, it assembles or invaginate into the ER, through which it will just go through the trans-Golgi network and get secreted. So this is basically with the help of the lipid droplet, which is also the product of high amount of the synthesis of the lipid. This is the reason that it needs the lipid droplet. The assembly of the virus happens close association on the lipid droplet and surrounding ER membrane. So that's basically the strategy, all different strategies. All right, so here also, this is another term that we use all throughout, but again, definitely triple underline this, that this is, we call it as a co-option. There are some membranes and the processes, and these are basically used for a different purpose by the virus. You will see that in the notes, I just put it in italicized, in italics, that what is basically the co-option. So definitely know the definition of that. So basically what they are trying to do, that you have this ER and how the different viruses are remodeling them, use it for a different purpose. That is your co-option. These are the different structures. I'm not describing that. You can just read that double membrane, single membrane vesicles that you have. But we have already seen the poliovirus basically replicate on the surface of these membranes. And some of them will invaginate and form these structures, double membrane structures, and it could be two different things. One of them, once they get like a single membrane, these endosomes will fuse with the lysosomes, and these lysosome and these proteins, they in the acidic endosome, the viral protein, which is if you remember that, in the capsid you have the VP1, VP2, VP3, and VP4, and VP0 is 2 and 4. So those needed to be cleaved, and those are processed in these vesicles. And once they are done in those vesicles, remember that the virus, the mature virus, can be released by, this is basically the exocytosis of the virus. These are releasing the naked virus when a single membrane is fused with the plasma membrane. On the other hand, we have also seen that this double membrane, if the virus is being formed in the double membrane structure, they ultimately fuse with the plasma membrane, and the single membrane structure, they can be release the virus inside these membranes as well. So there are several different things that can happen. So this is basically that how the remodeling of these ER membranes, basically for the virus release, viral protein synthesis, viral assembly, and release can happen basically. All right, so that's pretty much this chapter.